really what this unit is about. So this is just introductory stuff here. The cell repair process can follow several basic steps. So there's the first thing is there's interaction of the radiation with the DNA, which yields a DNA damage. So for example, so it could be direct or indirect damage. So the annual dose from background radiation, just annually, that a person would get, regardless whether or not exposed to a radiation source, is nearly 100 instances of DNA damage per cell per year. So this happens all the time. Only about 1% of these double-strand breaks, and most of the time the cell repairs itself error-free. So if you look at that, that's a lot of DNA damage done per year, and pretty much all of that gets repaired. When DNA damage is sensed, there's some molecules involved in the repair process. One is this AT molecule, and this protein stands for, senses double-strand breaks and initiates response. Some people suffer from that disease there, which I'm not going to attempt to pronounce. And so they actually have a very high likelihood of cancer development. Once a damage is sensed in the cell, it'll go into cell cycle arrest to allow for more repair time. So recall from last week, the cell's going through this process here. So the mitosis, the G1, the S, and the G2. Phase, and so what will happen is when damage is sensed, the cell cycle will stop to allow for repair. So there's cell cycle arrest. So there's different checkpoints in the cell cycle. So there's a checkpoint at the end of the G1 phase to assess if the cell is large enough to start the division process, the duplication of the DNA and the division, and it also senses if there's enough nutrients to commence the synthesis phase. For radiation damage, there's a checkpoint at the end of the G2, which is very important. So it checks to see that the cell is large enough to divide, and if all DNA has been faithfully duplicated at this point here. If DNA damage is sensed at this point, the cell will initiate a cell cycle arrest and respond to the damage. So the response to the damage can have different forms. The most important ones are apoptosis and repair. Apoptosis is a programmed cell death and mode of cell disintegration, so which results in the cell death. So it's characteristic for cells who sense that they're damaged and unable to repair. So the cell will basically die, or it's like a cell suicide, and that the idea of that is to prevent the DNA damage being passed on to future cell generations. So it's a very important mechanism. So it would rather have a cell die than basically mutate. There's a tumour suppressor gene, P53, involved in regulating this process, and so many tumours actually have mutated P53 genes which actually stop this apoptosis process. So let's have a look at some cell repair. Now this is only very, very brief and quick few seconds of something for which whole courses are designed to cover. So let's have a look very briefly. Single strand breaks, base damage and simple lesions. So repaired by basic excision. So basically we have a break here, and basically the cell chops out all of the bits around there, and then uses the we've got one strand down the bottom here. This strand still is okay, so it'll use this strand to duplicate the other side of the DNA and just basically paste it back in. So it's cut and paste. For there's double strand breaks, there's homologous recombination. So this occurs in the in that second checkpoint that I was talking about 2 slides ago. So this is the point in the cell cycle where there's actually 2 copies of the DNA available. So if one of the DNA molecules is damaged, the other DNA molecule can be used to copy across and do a repair of the damaged DNA. So this is slow and only available in the SMG2 phase. Then the other way is to, if it's non-homologous end joining, so it's independent of the cell cycle, and this is basically, it will just fit new ends, it'll just cut out the broken bit and fit new ends together. This is a more error-prone way of repairing damage, but it's responsible for most of the repair. Both double-strand repair mechanisms involve several steps and protein complexes. So the time required for repair depends on the type of lesion, the type of damage, the phase of the cell cycle that the cell is in, and the repair mechanism itself. So it can take a few minutes to several hours to repair, and this will impact on the effective time between irradiations. So we'll go into this a bit later on. So this will affect the fractionation of, for example, in radiotherapy treatments. All right. So this was a very brief, not intended to be at all rigorous, just a brief introduction to the cell cycles. So we'll move on to the next part of the lecture.